So a couple final things about the external versus internal factors that regulate the cell cycle. So external factors will actually trigger those internal factors. So those signals will come from the outside of the cell, okay, because the cell generally doesn't know the whole picture of the body. So it has to be controlled by something that can see how everything is kind of fitting together and working out. And that's where those external factors come in. So the external factors will trigger the internal factors, which are those cyclins. So they tell the cell which cyclins to produce, which of course then goes on to control what stage of the cell cycle that cell is a part of or in. So cyclins are created depending on the stage of the cell, depending on the stage of the cell cycle that cell is in. So all of that is controlled by your external factors. And those kinases are, again, activated based on which cyclin is expressed at that time. So the cyclins basically create the signal inside of the cell, and they do that by basically getting a signal from outside of the cell, but then the kinases are actually what go out and implement that signal and carry it out. All right, moving on into apoptosis. So we've talked about apoptosis briefly in this chapter, but again, apoptosis is programmed cell death. And I always remember a pop kind of sounds like the cell is popping itself, which is basically what it does. So it is actually a normal feature of a healthy organism. And we're going to see in a little bit what happens when apoptosis isn't triggered when it's supposed to be. Okay, so it is caused by a cell's production of self-destructive enzymes. So your cells do actually have a self-destruct sequence. Okay, and that can be triggered most often by some type of DNA damage or other type of cellular damage. It also occurs in the development of infants. Okay, so I don't know if you knew, but as you were developing as an infant, you actually had webbed fingers, okay, and toes. And so apoptosis actually works in that kind of webbing and takes it out. Okay, it's just easier to kind of grow that way and then take it out later. So every once in a while you will hear about a baby being born with webbing between their fingers or their toes, and generally that is a surgery that happens shortly after birth to correct that. And that's just because apoptosis was not triggered properly there. Okay, so again, a lot of times you think of cell death as being a bad thing, but apoptosis is actually a really good thing. It's kind of along the same idea of pruning a plant. Okay, you take off the dead portion of the stem in order to allow new growth, healthy growth to occur. So that's kind of the same idea of apoptosis here. Okay, so whenever apoptosis and regulation of the cell cycle fail, one of the side effects can actually be cancer. So cell division is uncontrolled in cancer. It's one of the main unifying characteristics of cancers. Okay, because most cancers, most of the time when you talk about cancer, generally a lot of people think of it as kind of one disease, when it's in reality thousands of different kinds of diseases. And cancer is usually named after where it was first discovered. Okay, that's why, breast cancer is found in breasts. You get throat cancer in the throat, lung cancer, so on and so forth. Cancer can then travel from where it originally started into different organs. Okay, that's how you can get basically breast cancer throughout the body. It started in breast tissue, but then moved from that location. Okay, so cancer cells will form disorganized clumps called tumors. These are generally incredibly dense because you have all of these cells dividing as fast as they can. And so there's no room for them to move out. So say you have enough room for 10 cells in this one area, but they're cancerous. And so instead you have 100 cells packed into that same volume. Okay, so they're very, very dense and packed in there. You do have two different major types of tumors. You have benign tumors, which remain clustered, and they can usually be removed through surgery. So we think of benign, oftentimes there is a misconception that benign means not harmful. Benign tumors can become harmful if they are not taken care of. It's just at that time, they are not causing the most damage that they have the potential to cause. Okay. Then you have malignant tumors. So these do something called metastasize, and that's where they break away. So on the edges of the tumor, you'll get the metastasis. Sorry, if you can hear that snoring, that's Potter. He's a deep breather when he's asleep. But the metastasis, the metastasizing at the edge of the tumor just sends kind of cancerous cells out from the tumor. Generally, they can make their way into a blood vessel and then basically transfer themselves to a different part of the body. And that's when you get issues, because then you have cancer popping up throughout the body. Okay? And you can, like, one cancer cell can go on to form a tumor elsewhere in the body. And that's where the issue is. Okay, so this is kind of looking at a different depiction of it. So we have our normal cells, as you can see all around, and then the cancer cell they made kind of purplish around the edges. Okay, you also see that there is a bloodstream nearby. So this is when the first cell becomes cancerous. Okay, so it's got some kind of cell damage, apoptosis has been turned off, it's not listening to the cell cycle regulation signals. It's just going at its own speed, and its speed is pretty fast. So it starts to divide really, really quickly. Okay? And so it very quickly outpaces the division of the normal healthy cells. And this is when you get the formation of a tumor. Now, as that tumor grows, it eventually becomes metastasized. Now, like I said, with the benign tumors, generally they have very solid, very defined edges to them. When it's metastasized, oftentimes the edges are more frayed, okay, which makes them even more difficult to take out. Because if you don't take out every single cancer cell, you haven't destroyed the cancer. You've just kind of postponed it and pushed it off a little bit. But in this final image, you can actually see some of the cancerous cells making their way into the bloodstream. So that is a sign that this has metastasized and the cancer is being transported to another region of the body. Okay. Now, generally, if you have a patient who has cancer, they say, you know, once they've gone through the treatment regimen and everything, and they are no longer showing signs of cancer, they're said to be in remission. Okay, that just means that they've had cancer, but they don't currently have any symptoms or any tumors. Okay, now generally the patient will be in remission for 5 years. Okay, if they make it to the end of the 5 years with no new tumor growth, they say that they have beaten the cancer. Thank you, Potter, for your commentary. If within those 5 years, if a tumor reforms, then they have relapsed and the cancer has come back. Basically, if they relapse, it means that they didn't get all of the cancer cells, that at least one was left somewhere in the body, and it reformed a cancerous tumor. If the patient has completely gotten through those 5 years with no reoccurrence, no relapse, then it means that they can say with at least relatively good certainty that they got all of the cancer. Okay? And then they have technically been cured of that type of cancer. Now, saying that they've been cured, they are still susceptible to that cancer. But, all right. So cancer cells are really problematic because all they're concerned about is dividing. and multiplying and growing and then transferring elsewhere. They do not carry out necessary cell functions, which is why you start getting different symptoms that are usually the first sign of cancer. So cancerous growth, like I said, forms kind of a mass, and they don't really care what else is around them. They just grow. So you can even get tumors, like tumor growth, in a dish in a lab, okay? Because they don't really care if they're touching another cell. They're still going to divide because they want to. So cancer cells come from normal cells that have damage to the genetic information in the cell cycle regulation signal pathway. Okay, so maybe that is a mutation in the off switch of a particular protein that stimulates cell division. Okay, so basically that protein is stuck in the on position and it's not allowed to turn off. So it's constantly pushing for more cell division. Okay. Now, cancer cells also take up more nutrients than normal cells. And this is where another issue with cancer comes in, where they're taking all of the normal nutrients from the healthy cells in order to basically fund their habit of cell division. Okay, so they start to starve the cells around them. And then they also can force the growth of more blood vessels in order to get those nutrients. So they will basically force blood vessels to grow to them in order for them to get those nutrients. And then that just makes it easier for them to eventually jump into the bloodstream and travel somewhere else in the body. So with cancer, we also have to talk about carcinogens. Okay, so carcinogens are substances that are known to promote the damage to genetic information that causes cancer. So this is where, cigarette smoke comes in. Okay, there have been some major concerns with even Juuls or, e-cigs, talking about all of that, especially because they're relatively new, so we haven't seen long-term effects yet. And those are just now starting to come in. But carcinogens, there are lists of carcinogens. They are separated out by kind of how dangerous they are. And they're constantly adding new carcinogens to the list and moving other ones around and everything. And basically, if it's one of the highest levels, then it's really bad. But the other levels, there's a lot of wiggle room in them, but it's still something to look out for. Standard cancer treatments typically work by killing both cancerous and healthy cells. They are not targeted to just the cancerous cells. That is actually a major point of research at this point, is how do you target these cancer treatments to just the cancer cells and not damage the healthy cells around them. So a lot of the side effects of cancer treatments are actually from the medications because you're killing off a good portion of your healthy cells in that area, and that's obviously automatically going to make you feel really bad and very sick, and it's basically a radiation sickness. So generally they will target cell division. Okay, obviously that's going to affect healthy cells as well because they do need to divide, but the cancer cells are dividing much more frequently than the normal cells, so it'll harm the cancer cells more than the normal healthy cells. Okay, and then these are the videos that I've posted. There are several talking more about why we haven't cured cancer, some of the myths about them, and then a couple different strategies that they're using to both target the cancer cells specifically and also new kind of theories of how to treat cancer. Okay, so there you go.